Hi, we're going to talk about sickle cell as a phenomena study for genetic expression or gene expression. So let's dive right in. So we're going to talk about sickle cell anemia. Sickle cell disease is a genetic disorder characterized by the abnormal shape of red blood cells, and that can lead to serious health issues. So here we have some beautifully contrasted red blood cells under the microscope. And if you notice, they're not all looking that healthy, beautiful, rounded shape. One thing to remember as we talk about this is genetics don't act in isolation. So even though sickle cell anemia comes from the same kind of DNA change, not everyone who has it will have the same experience. So some people are going to have more symptoms than others, and that's because people with sickle cell make an abnormal protein called hemoglobin S. But hemoglobin S can be in different amounts or ratios, and some individuals might have only a little bit of symptoms because their body is producing more healthy hemoglobin than unhealthy hemoglobin or abnormal hemoglobin. And that can also be vice versa, where some people experience many problems, and it's because their bodies produce less of the normal hemoglobin and instead create far more of the abnormal hemoglobin S. So let's talk about this from DNA to protein. So hemoglobin is a genetic blueprint that we're going to be looking for. The hemoglobin gene plays a crucial role in determining the shape of red blood cells. The form is how we get to the function. So red blood cells should be rounded so that they can flow easily through narrow passages, and they should also be flexible, not just for flow purposes, but also their structure, their form helps determine how well they can carry gases like oxygen. Normally, the messenger RNA sequence is going to be coding for glutamic acid, GAG. And this is a hydrophilic amino acid that results in hemoglobin folding into a beautiful, rounded, proper shaped red blood cell. So the sequence is crucial for maintaining the cell's flexibility and its function. A mutated mRNA or messenger RNA sequence is going to code for valine, GUG, and that causes the protein structure to misfold and it forms a stiff or sickle shape, and that impacts its functionality. That sickle shape gets stuck on each other, so this red blood cells end up sticking to each other. They don't flow as easily through the capillaries, blood vessels, etc. And they also don't carry oxygen as efficiently. And this is what happens when a single codon happens to have a change at the nitrogen base pair. This is just one error, one point mutation, causes a lot of problems for individuals with sickle cell anemia and sickle cell disease. So normal, healthy hemoglobin is going to be rounded and smooth, and it allows the red blood cells to flow smoothly through all of your blood vessels, arteries, etc. And it also allows for optimal oxygen transport throughout your body. Here you can see that beautiful rounded form. Whereas sickle cell hemoglobin is a more problematic shape. It's stiff, crescent shape, it sticks to each other, it catches on each other with those very rigid, pointed ends, and it causes a lot of health problems. It causes pain, physical in the body, and it also doesn't deliver oxygen as efficiently, and that can lead to your increased risk of infection or various other issues. Here we can see that example of that rigid, stiff crescent shape. So let's talk about some of how this can be described. So So the DNA structure, your genes as information. The hemoglobin gene specifically is on chromosome 11, and that determines the structure of hemoglobin proteins in red blood cells. Mutations in this gene are going to lead to significant changes in red blood cell shape, which impact the overall health and function of the whole organism, but also of that individual red blood cell. Protein synthesis. Well, we know that ribosomes are where proteins are made at. Your messenger RNA is going to have the codons, and those codons are going to give the information to code for specific amino acids. So during protein synthesis, the hemoglobin gene is transcribed into mRNA, which is then translated into the hemoglobin protein. So the hemoglobin gene is transcribed in the nucleus, and it's transcribed into messenger RNA. Messenger RNA can exit the nucleus and then is translated at the ribosome into the hemoglobin protein. Mutations. Well, there's different types of mutations. Sickle cell disease is caused by a specific mutation in the hemoglobin gene, and that's known as a point mutation. It replaces the codon for GAG with GUG. Gene expression. So from protein to phenotype. Well, sickle cell is a phenotype. We can classify it. It's a physical type of change that we can classify by looking at the red blood cells of an individual who happens to have it. Changes in the protein structure, having that point mutation, causes a change in how the protein's shape is constructed. And that shape change is going to impact the functionality. Genetic variation. So we're just going to touch briefly on this concept, but I think it's important and vital to talk about when we're talking about sickle cell disease. How did a sickle cell disease mutation get into the genetic inheritance of a population? Well, there has to be a reason for it. So understanding sickle cell, part of that understanding also comes into play with natural selection as well. So let's talk more about that. So there's a connection to malaria. Malaria is caused by a parasite that is transmitted by mosquito bites. So malaria is caused by a parasite that infects red blood cells. The parasite grows best inside normal round blood cells. So if you don't have normal and round blood cells, you're not as prone to getting malaria as somebody with rounded normal red blood cells. So the malaria parasite can't live as easily in the sickle rigid shaped cells of somebody with sickle cell disease or sickle cell trait. So let's talk a little bit about these differences. So sickle cell trait versus sickle cell disease. And remember, we already talked about how genetics are not acting alone. So normal, no sickle cell genes are going to be described as HBAA. And we'll talk more about what that means later in the year. But right now, that means all red blood cells are normal and you can get malaria very easily. Sickle cell trait is going to be coded HBAS, half normal, half sickle cell. And these are going to cause mild or very limited or no symptoms. But you're protected from malaria. You're not going to get as sick because some of your cells don't have that rounded shape. Now, problems come up when you have sickle cell disease, and that's HBSS. And all sickle cells, so all of your red blood cells are sickled shape. And that causes serious health problems because that sickle shape is not very good for transporting oxygen or flowing through your arteries and veins and capillaries. So how did this happen? Well, some of the reason it happened is, if we look at this map, this is the continent Africa. And if you notice, there's a lot of malaria-carrying mosquitoes in the midsection of Africa. And this is going to cause some serious problems because anybody living in this area will have exposure to malaria. So sickle cell disease serves as a powerful example of how a single genetic mutation can lead to significant changes in physical traits and health outcomes. When we talk about natural selection, we will circle back to that dominant malarial vector species in Africa and how individuals from those areas are going to pass on their genetic traits, including the sickle cell trait, because it helped keep some ancestors alive, but it did kill off some individuals. But we maintain that in our genetic code in any individual who survived from that area. So malaria, very interesting to talk about and how it impacted a whole continent. So I can't wait to talk more about this with all of you. Bye.